Hi, my name is Melissa Andrews, and I'm a lecturer in systems physiology here in biological sciences at the University of Southampton. I sit within the sort of neuroscience research group in the School of Biological Sciences, and my main research focus is in terms of regeneration of the nervous system, axon regeneration, and repair. What I'm going to talk to you about today is a project that we have regarding stem cells and attempts to repair the brain using those stem cells. But initially, just a little bit of background. Here we can see a picture of Christopher Reeve along with his son. Christopher Reeve passed away several years ago, but he was sort of he was the chairman of the Christopher Reeve Foundation, but he brought about a new sort of awareness to spinal cord injury. And spinal cord injury is what I'm mainly interested in repairing paraplegia and that sort of thing. But here's just a small tribute to him and the sort of awareness he brought to the condition that I work on. Now, in terms of spinal cord injury, you can see that there's a wide range of causes of spinal cord injury, and here's just a few. Motor vehicle accidents are predominant among the causes, but sport injuries such as skiing, unfortunately, horseback riding, DIY, do-it-yourself jobs. As you can see, the man on the bottom right is climbing onto his roof from a ladder, very dangerous, and in the sort of recent years this has come up as a more prominent cause of spinal cord injury, unfortunately. So we all have to be careful. Now, I put a small picture here of just a skull and vertebral column, and I question, is that protection? Yes. We are not meant to damage our brains and spinal cords because we have this bone that encases both the brain and the spinal cord. But as we know, because of all the activities we do, this protection may not be enough. Yes, we can wear a helmet in terms of motorcycle riding, bicycle riding, but again, spinal cord injuries still happen. It mainly occurs in developed countries. As you can imagine, those sort of sports and motor vehicle accidents are occurring much more in a developed country than in an undeveloped or third world nation. But across the world, paralysis is actually starting to happen with more abundance. Now, here I'm just sort of differentiating down the different types of injury that someone can have, as well as the different symptoms that someone can have. So if you look at the sort of top half of the page, if an injury occurs in the neck region, the cervical or cervical region, you're likely to have what's called quadriplegia. So you then are unable to move your arms as well as your legs. So if it's very high up in the neck, very high up in the cervical region, you may then also be ventilator dependent. So you wouldn't be able to move your lungs on your own and the sort of muscles surrounding your lungs. But if that injury is a few levels down in your spinal cord, so C4 would be the first injury, that's high up in the neck, C6 would be lower down, you may then not be ventilator dependent. So you have a little bit more independence, yet you still are quadriplegic and you can't move your arms and your limbs. Now, this is sort of contrasted with a paraplegic person. Their injury has occurred much lower down in their spinal cord. So the sort of third scenario down is a T6 injury. This is happening in the middle of the thoracic cage. So if you're in the thoracic cage and if your injury is in the thoracic cage, you actually now have paraplegia, or you have a chance of having paraplegia. Your arm control is fine. You just now can't control your lower limbs, your legs, and you likely have reduced function, reduced sensation in your abdomen and pelvis. And then in the fourth scenario, this is L1 injury, that's your lumbar area. This is the very lower part of your spinal cord, or towards the lower end of your spinal cord. In this case, if you injure that area, you likely are just having problems with leg function. You might have spared the abdomen control and the sort of sensation around the abdomen and pelvis, and you're now just looking at paraplegia of the lower limbs only. So it's a very heterogeneous condition, spinal cord injury. There's no two injuries that are alike. It can vary depending on severity as well as where it is in the spinal cord. damaged spinal cord. I have a picture of a hand here, that's not a spinal cord. But what I want to talk about and introduce here is the fact that our body has the ability to repair under certain circumstances. So in the case where you have a cut on your hand, this person has, minor treatment is needed. A plaster, a bandage, possibly some sutures or some stitches. And you end up with a small, or even not even a scar at all. Our skin has a great capacity for repair and regeneration. Now contrast this with our nervous system, our brain, our spinal cord. We have a very reduced capacity, if at all, to repair and regenerate. Now in this cartoon drawing, this is a spinal cord that's been laid out horizontally, and it's a cartoon to sort of depict all the massive processes that happen. So we have an immune response, we have a lot of cells that sort of wall off the lesion. It's our body's way of trying to protect the rest of the nervous system for the lesion spreading. But what it leaves behind is an inhibitory environment. Walled off cell areas where new cells can't grow through. You even have holes in the tissue we call cysts. And it's actually a formidable barrier that, results in an inability for the brain and the spinal cord to regenerate and repair itself. So in my lab, what I'm interested in is something called an integrin. Now an integrin is, we can see a picture on the bottom of the slide, it's a transmembrane receptor. All that means is it's located on cells, and it has an outside of the cell domain, an extracellular domain, and it also has an inside of the cell domain, an intracellular domain. So it's a big protein that sits on the cell surface, but it sort of translates or communicates between the outside of the cell and the inside of the cell. We have them in all cells of our body except for red blood cells. But the interesting thing about integrins in the nervous system, they're found in high abundance in a developing nervous system because they help our nerve fibers find where they need to go. They help our nerve fibers extend from the brain down to our limbs, etc. But once maturity of the nervous system happens, these integrins disappear, they're downregulated, they're no longer needed. And then upon injury, they don't come back. So we're missing a vital protein that's very useful in development, and that, we can't use in terms of repair. So some of the studies that I've been involved in is looking at what happens if we reintroduce these integrins. So in these two slides that you can see up at the top, these are, they're neurons that have been grown on what we call an inhibitory substrate. So a substrate, an extracellular matrix that you would find in an injury. And what we want to see is we want these cells to put out nice long nerve fibers, long processes, indicative of, successful growth. And what you can see with the arrows is that they don't really put out long processes. They put out these little stubs and then they stop because the inhibitory protein doesn't allow for much growth to happen. And these cells don't have integrins. Now if we put integrin into the cell via means of transfection or transduction, so we add this sort of DNA to the cell in order to get them to express the integrin. Now we're looking at a specific integrin type now, this is alpha-9 integrin, but that's neither here nor there. But what you can see in the arrows, or with the arrows highlighted, is these very long processes. So in a way we've sort of reversed this inhibitory effect, and we've gotten our cells, our nerve cells, to grow on an inhibitory substrate just by bringing back a molecule that was found in development and is now, we put it back into an adult situation, or these are adult neurons, and we get growth again. This is under cell culture conditions, so we don't have all the inhibitory molecules that we saw on that busy slide before, but it serves a sort of case in point to say that we can recover some growth. Now, we've taken this study even further to say, okay, now if we bring back our integrin of choice, which is that alpha-9 integrin, and we also bring in an integrin activator, that's kinlin-1, we can actually induce a significant amount of regrowth. And one of the projects that we've been involved in and published a couple of years ago is this project here. And here's just a snapshot of one of the results figures. We looked at sensory fibers. We looked at sensory nerve fibers, and we injured them. Then we reintroduced our integrin of choice, the alpha-9. We reintroduced an integrin activator. So we introduced the protein and we got it in its active state. And what we found was when we injured, and you can see sort of the schematic on the very right-hand side, the sensory nerve fibers that control the hand, so this is in the sort of the lower part of the neck, the lower part of the cervical region, so we injured those nerve fibers, and we got axons, or we got nerve fibers, to regrow back up into the sort of high neck region where they would normally grow before they extend up into the brain. What we found here was a significant amount of regrowth. Sensory nerve fibers, they tend to grow much better than their motor nerve fiber counterparts. So you can recover pain sensation much more than you can recover the ability to use your hand, but that's neither here nor there. The amount of regrowth that we saw with this integrin reintroduction and its activator was more significant than anyone has seen in the sensory nerve fiber regenerative field. Now, that's one of the projects that we've been working on and published on a couple of years ago, but another project that we've been involved in is using stem cells to replace neurons. Now, there's several stem cell types in the world and being used in research, but the one that I'm going to talk about and the one that we use in our lab are stem cells called induced pluripotent stem cells. And if you see this sort of diagram at the bottom of the slide, what you can see is, well, an induced pluripotent stem cell can be from any cell of the body. So it can be a skin cell. We take that skin cell, we de-differentiate it in cell culture in that first dish, and then we throw a mixture of factors on it, and we can continue those factors even further, and we can make that skin cell into any cell type of the body. And then I can just say we can almost make a new person. That's not being done because that's not the point of using stem cells. But what is the point is to see whether or not these stem cells have the ability to regrow things like our nerve cells in our brain and spinal cord after injury, which don't have their own means of regrowing unless we put in things like integrins. So a project that we've been doing and we published on a couple of years ago involves implanting or transplanting these stem cells into a rodent brain. Now, these stem cells are human stem cells, so we're looking at putting a human cell into a rodent brain to see what actually happens. And in this particular scenario, we're looking to see if, now in this sort of right-hand side of the screen, you can see these this long nerve fiber, and the green just depicts the integrins. So in this particular scenario, our integrins are expressed because in these stem cells, well, no holes barred. Everything is sort of in a developmental state. So we have our integrins already there. Our cells are primed to grow across an inhibitory substrate. Now, the first experiment that we've done is we've just transplanted human cells into a rodent brain. There's no injury in this case. We just looked to see whether or not we could get these stem cells to project out nerve fibers alongside the nerve fibers that are already in place. And in fact, we were able to do that. And what you can see here is you're looking at sort of a sagittal view of how these nerve fibers basically traject from where they were transplanted in that sort of purple area on the sort of left hand of the screen, and the red, those little fibers, well, those are axons or nerve fibers that have come from our stem cells. And what we can see is they integrate well, they survive. This is almost 2 months after transplantation, and we're able to get nerve fibers growing alongside the fibers that are already there. So you can imagine that if this were in a regenerative scenario where there's an injury, these nerve fibers and these stem cells could produce fibers to grow through the injury. And again, remember, these stem cells are primed. They have all of the growth promoting molecules that you would want, such as integrins. Now in this particular slide, we've taken the tissue that you saw on the previous slide and we've done some 2-photon imaging. So this is in collaboration with a group up in University of St. Andrews, but what they've done is they've sort of just taken the nerve fibers and their fluorescence intensity and looked from all angles, and you can see that there is an abundance and quite a nice sort of thickness of these nerve fibers. So they're quite healthy and they're growing basically around the tissue that you can't actually see in this point because we're just seeing the axons, but it was a tremendous amount of regrowth that we saw, well, new growth rather, with these stem cells. Now in terms of an undergraduate project, what do I offer in my own lab? Well, I have a few examples here. Basically my projects are either cell culture based or they're tissue-based, where a student would then sort of look at some tissue that was produced by myself or a postdoc in the lab. And in the first example at the top, it's quite a wordy title, but we were looking at reintroducing our integrin and seeing whether if we broke down some of the extracellular matrix, making it less inhibitory, if we got a better expression of our integrin. So it was more of a technical question, but the student that worked on this had some very nice results. She's now doing postgraduate medicine at Bart's, and she did very well in the project. The project at the bottom, the student is now doing a PhD over in Bath, and she was doing a cell culture-based project where she was manipulating the integrins to get them to transport in the nerve fiber better, to be expressed better. And she did very well in that project as well. So all in all, I do a range of projects. We're always interested in the question of, how can integrins help our injured nervous system? What can stem cells do? But in the end, it's about enhancing regeneration. We do it.